Cottingham, and welcome to this news briefing from the 252nd National Meeting and Exposition of the American Chemical Society in Philadelphia. We're joined today by Drs. Christopher Bettinger and Hong-Ah Park from Carnegie Mellon University. They will be talking to us about a battery you can swallow that could power future edible medical devices. Dr. Bettinger?
Katie, thank you so much for having me. So it's my pleasure to discuss our discovery and innovation in the world of polymers as they apply to batteries for ingestible medical devices. And so really what we're talking about here today is the notion that you can use biologically derived materials as important components of an ingestible battery. And so our broader vision for this work is really thinking about batteries as they exist today and making them application specific for edible electronics. And so specifically what we've discovered is that we can use melanin pigments. These are the same pigments in your hair, in your skin, and your eyes as components for these ingestible batteries. And so these have significant advantages over existing batteries because they're designed for this purpose. So we've basically one by one replaced the potentially toxic components of a battery with that of benign endogenous materials within the human body. And so at this specific meeting, what we're reporting is essentially a very specific dependence of the performance of these batteries as a function of the ion within the body. So we've previously shown that we can use things like sodium to replace lithium and other kinds of ions, but here we're exploring other kinds of ions like aluminum and iron. And what we've really discovered is this is a way we can not only think about new kinds of cations, but also then learn something about the biology of melanin as a pigment.
Okay, Hong-Ah?
Yeah. I'm working on melanin batteries with different monovalent ions from lithium, sodium, potassium, rubidium, cesium. Those cations have different size and different properties, but chemical groups and fulfilling structure of the melanins have bound with those cations, and melanin show higher specific capacity for those cations. And we are studying this chemical structure of the melanin after bonding with those cations. Yeah.
Okay, did you want to show the prop you brought?
Yep.
So there's the ingestible battery?
Yes. So this is a capsule with battery. So we open the capsule. So
and what you're seeing there is essentially a component that's actually 3D printed and degradable, and it's encapsulated in a gelatin material that would be similar to what you might see in a vitamin, for example. And so within that, there are the kind of the functional parts of the battery. And so these are melanin electrodes. Again, so this is essentially repurposing a naturally derived pigment as an important component of a battery. And so another important part of this design is actually the electrolyte. So a lot of times an electrolyte in batteries that power your cell phone, your laptop, they're very exotic and they're made of potentially toxic materials. But in this specific design, what we're utilizing is an aqueous electrolyte, namely the fluid in your GI tract. So we think that's another kind of way to capitalize on the endogenous material in the body to have a functioning battery.
Okay, excuse me, any questions? Please remember to state your name and affiliation when you're asking a question and wait for the microphone.
Hello, Ben Valsler from Chemistry World. Sorry I missed the very beginning here. There are a range of different ingestible devices that people are using now. Do you see any limitations in the use of this battery, or do you think that they could go in any of the existing technology we already have?
Yeah, so that's a great question. So we think about that from kind of almost like a risk perspective. And so for sure, there are existing devices like ingestible cameras that use what's called off-the-shelf battery materials. These are the same batteries you might find in like a hearing aid or a watch, right? And so we think about this as those aren't really designed specifically for this new kind of application. in fact, they sometimes can pose a significant risk to the patient, especially after repeated, let's say, events of dosing, right? And so we think about this as a way to sort of de-risk those kinds of batteries. And so, so they're cost-effective, they're non-toxic, and they have a performance that's completely adequate with this application space. And we can just talk a little bit more about that. So they can provide about 10 milliwatts of power, and that's enough to power really any kind of existing medical devices. Most other kinds of like energy harvesting techniques or glucose oxidase fuel cells are going to be like microwatts of power, so we're 3 orders of magnitude higher than those kinds of kind of energy sources. The other thing is that they don't last for very long, so they last for about 10 to 20 hours, and so in terms of an actual battery of powering a computer or a laptop, they're actually quite poorly performing. But kind of the trick is that we think an ingestible device would only have a lifetime of about that. So if you just ate lunch or dinner, the food's going to last about 10 to 20 hours in your GI tract. And so we kind of have a harmony between the timescales of operation between these kinds of batteries and the kinds of timescales for device operation in vivo.
And you said it was completely degradable as well. If something goes wrong with the gelatin capsule and we end up digesting the battery rather than allowing it to degrade, what does it break down into? And do we know that those components are innocuous?
Yeah, so that's a great question. So there's kind of a couple of different levels of degradability. There's kind of like a mechanical degradability, so we can have kind of mechanical disruption of these devices, and there's kind of chemical breakdowns of these materials. And one thing I'll offer is that the same kind of pigments that we use are actually the same kind of pigments that you find in a squid ink pasta, actually. So if you ever had squid ink pasta, you've already consumed more melanin that are in the batteries that we're going to be talking about this week.
Thank you very much.
Bela Buslig, Office of Public Affairs, ACS. These batteries, apparently, at least the way I visualize it, is one particular kind of ion on one side, one other kind of ion on the other side, and both supported by melanin. And what's separating them, the two electrodes? And if you need higher voltages, how do you stack them?
Yeah. Did you want to talk about that, Hanga?
So we are using pectin, which is chemical polymer, but we also found the pectin in jelly. So by using those material as a separator, you can make the battery eatable and ingestible for medical device applications.
Yeah, so just to build on that, the separator is actually the simplest thing, right? So basically any kind of carbohydrate could potentially satisfy the qualifications of ingestible and electrically insulating in this context.
Now, how do you control the degradation of the housing?
Yeah.
Because, everything you swallow ends up in the stomach,
yeah,
and probably more hydrochloric acid than you'd ever want to put on anything, anything that you eat.
That's right.
So how do you prevent the hydrochloric acid from eating up the whole thing, or shorting out the battery?
Right. So, so I want to come back to a previous question you had about the kind of design of the system. And so melanins are going to be operating as one part of the battery, either the cathode, the positive terminal, or the anode, the negative terminal. It actually works well for both. Now, the other side has to be engineered appropriately. So the first thing is that the melanin is only half of the battery. And the second question that you pointed out very astutely is, what about the stability of melanins in these kind of very caustic environments? And so it's counterintuitive, but melanins are actually very stable in acidic environments. They become less stable in basic environments, and that's kind of related to their redox chemistry. And so actually in natural melanin is actually synthesized in little melanosomes where the pH is actually around 3 or so. So it's counterintuitive, but it actually becomes very stable. So now kind of back to the degradation idea. So that's why we talk about kind of mechanical degradation. So these are basically laminates of nanoparticles. And so while the melanins aren't going to chemically degrade very rapidly, they are going to be mechanically sort of just perturbed by the mastications in the GI tract. And so then that kind of mechanical insult will break them down, and then they'll make smaller pieces, which can then transit the GI tract. And that's only in the case of retention.
Now, how do you control the outer casing of the thing? Like it goes from a very acid to a very alkaline environment when it's in your stomach or in your intestines.
That's right. So there's this very chaotic sort of balance of pH environments, right? So pH of 1 up to pH of 6, they can kind of right. And so how do you manage that? And so we basically leverage a lot of the materials that are already developed for other kinds of medical devices, like PLGA, for example. So a lot of it's known about how it degrades in response to different pHs. And so in principle, a device could be engineered with that previous foresight and knowledge, right? And so you could think about basically two different timescales: a timescale for transit and a timescale for reasonable disintegration. So you could design a capsule that would be intact for, let's say, on the order of 3 days, right? And so it would be stably operating. And in the unlikely event that it gets retained, then it would kind of engage this other process of degradation and disintegration, and then would then harmlessly break apart into small pieces that can transit the GI tract. So the question about timescales is very important, and it's very nuanced, but I think that would be an important consideration in a prospective device that would utilize these kinds of electrochemical storage systems.
Thank you.
Okay, we have an online question from Christine Sa, Office of Public Affairs. She says, some scientists have been known to test their inventions on themselves. Have you swallowed your batteries? And we do see a picture of you with the battery on your tongue.
That's right.
So have you actually ingested it?
Yeah, I actually have. So
and you're still alive.
That's right. I'm still here. I'm still cogent. Good. So I think there's a very interesting kind of thread that's kind of wrapped into that question too, and it's basically on the evasiveness, right? So I have never in my career tested an implantable material on myself, but I've already tested an ingestible material. And I think that speaks to our compliance with oral medications and oral systems as opposed to implantable devices. And so I think that's a really cool question. And so short answer, yes.
Okay. And I was wondering, how did you get the idea to use melanin of all things?
Yeah, so that's now back to the basic science. So melanin has a really interesting function in the body. It's mainly protective, right? So we usually think about melanins as a material that can absorb UV light, right? So we get more pigmentation on our skin, and that's great. It also is responsible for scavenging free radicals. So these are kind of spare electrons in the environment, and so it can do that very efficiently. And so it turns out that that's one important process, right? So it can kind of exchange electrons very easily. That's also important in a battery, right? But the melanins also have this really interesting property in that they are found actually inside the brain, in the midbrain, right? So the question is, why is there melanin in a place where there's no light, right? And so it turns out that melanins in that context are really designed to kind of chelate or grab onto toxic metals. And these are things like aluminum and iron and zinc. And so we thought, okay, if they have this kind of electron exchange capability, and they have this kind of cation chelation capability, then that's really what a battery material is in its essence. So we've really just leveraged those existing properties in a different kind of context, and then from that we have this kind of this new invention.
Okay. And are you looking for industry partners to collaborate with, and how long will it take to get to market?
Yeah, so I think that's a great question. I think it depends upon the specific indication or the specific technology. We think there is merit in a drug delivery system in terms of, let's say, a smart pill, and something where you could take a pill every single day, and we're reducing the risk of an ingestible battery is sort of prudent, right? So, for example, it could be a pill that delivers a certain drug to a certain location in the GI tract to maximize the absorption of that small molecule, or it could be a smart pill that can deliver a payload of bacteria to a certain part of the GI tract to locally modulate the composition of the flora. So those are, we think, kind of logical next steps, and we're definitely looking for partnerships for to sort of advance this technology in that context.
Okay. Other questions? Okay, thank you very much for joining us. The archived version of this session will soon be posted at bit.ly/ACS Live Philadelphia. Please join us for our next press conference tomorrow at 9 a.m. on the development of an oral insulin pill for treating diabetes. Thank you.
Thanks.